Hello everyone, my name is Deicy Vega. Thank you for coming in today for my presentation. Today I'll be talking about the effects of Astaxanthin treatment on a rat model of Alzheimer's. There are two reasons why I chose this topic. The first reason was because I wanted to see how much closer has science gotten us to a cure for Alzheimer's disease. And the second reason was that I chose this topic due to the lack of knowledge within my community regarding Alzheimer's. I wanted to get educated regarding the disease in order to spread the information within my community so that they can keep an eye out for the symptoms within their loved ones. This way, they will have a possibility of detecting it in the early stages, which will better prepare them when they have to discuss it with their provider and make informed choices about their treatment plan. An outline of today's project, I will be giving you a little background of what is Alzheimer's disease, the experimental methods of how scientists use lab rats in order to get us closer to a cure, the data collected along with the results, and the take-home messages from the overall presentation. There will also be a page that will be dedicated for the resources that made this presentation possible. So, Alzheimer's disease. Um, Alzheimer's disease is also known as AD. It is a progressive neurological disorder that causes the brain to shrink and brain cells to die. Alzheimer's disease is the most common cause of dementia, a continuous decline in thinking, behavioral, and social skills that affects a person's ability to function independently. AD typically destroys the neurons and their connections in part of the brain involved in memory, including the extrasensory cortex and the hippocampus. It later affects the areas in the cerebral cortex responsible for language, reasoning, and social behavior. Symptoms include, but are not limited to, memory loss that disrupts daily life, challenges in planning or solving problems, difficulty compelling familiar tasks, confusing with time or place, trouble understanding visual images, misplacing things and losing the ability to retrace steps, decreasing or poor judgment, along with withdrawal from work and social activities, and changes in the mood and personality. There are many misguiding myths there's in the community that I would like to address and correct. The first myth is that memory loss is a natural part of aging. In reality, as people age, it is normal to have occasional memory problems, such as forgetting the name of a person you recently met or where you've placed your keys. However, Alzheimer's disease is more than occasional memory loss. It's a disease that causes brain cells to malfunction and ultimately die. When this happens, an individual may forget the name of a longtime friend or what road to take to return home after where they have lived for decades. The second myth is that older people can only get Alzheimer's. Alzheimer's can strike people in their 30s, 40s, and even 50s. This is called younger onset Alzheimer's, also referred as to early onset. There are currently 200,000 diagnosed patients younger than the age of 65 with onset Alzheimer's disease. The third myth is that Alzheimer's disease is not fatal. As of now, there has been no survivors with Alzheimer's disease. It destroys brain cells that can cause memory changes, erratic behavior, loss of body functions. It slowly and painfully takes away a person's identity, ability to connect with others, think, eat, talk, walk, and find his or her way back home. According to the Alzheimer's Association, located in the United States, an estimate of 6.2 million Americans aged 65 and older are living with Alzheimer's dementia as of 2020. 1 out of 9 people aged 65 and older would be detected with Alzheimer's. Unfortunately, two-thirds of Americans with Alzheimer's are women. There has been no linkage of why most of the patients affected are women, but scientists are looking into it. In 2021, Alzheimer's will cost the nation an estimate of $355 billion. The cost is also estimated to increase as high as $1.1 trillion by 2050. The reason for such a high cost is due to the burden and the time spent on caregiving for patients by caregivers. Specialty providers such as the neurologists, physical therapists, and others that may be involved in the treatment plan. And also the price tends to increase when the progression of the Alzheimer's disease evolves and gets worse over the time. Another additional cost would be the prices of the drug treatments, as they can be very heavy. It is important to come up with a solution to this problem. The patients deserve a fighting chance and a better quality of life. The experimental hypothesis was as follows. The effect of oxidative stress had been recognized as the main contributing factor in late-onset Alzheimer's disease. The potential of astaxanthin in treating Alzheimer's disease by investigating the effectiveness of astaxanthin in Fab-induced rats. If Fab-induced rats are induced with higher dosage of astaxanthin, then the Fab-induced rats will display less symptoms of Alzheimer's disease and be able to complete the Morris-Water maze. Astaxanthin, also known as ATX, has potential of reducing the oxidative stress due to its antioxidants. That is why this is one of the things that the scientists wanted to study with, because they saw hope. The material for this experiment are as followed. They needed a total of 60 ml Sprague-Dawley rats, also known as SD rats. They needed the ferrous amyloid buthenine solution, also known as FAB solution. They needed astaxanthin treatment, also known as ATX treatment, and a Morris-Water maze. For the SD rats, they had to be 3 to 4 months. The reason why scientists selected this kind of rats was due to research showing that humans and SD rats are remarkably similar in structure and function within the brain. Both consist of a vast amount of highly connected neurons that are constantly talking to each other. The FAB solution, when injected, it mimics the AD-like pathology. Therefore, when injected on a rat, it makes the rat have the same or similar symptoms as those patients with the Alzheimer's disease. And the ATX is the oxidant that protects against oxidative stress, variable disease, and age-related degenerations. I will be referencing the ferrous amyloid buthenine as FAB, as it's easier to say, as well as for the astaxanthin, I will be referencing it as ATX. Last but not least, the Morris-Water maze is designed to test spatial memory and long-term memory by observing and recording escape latency, distance moved, and velocity during the time spent in the maze while trying to escape. The scientists wanted to create the same environment for all the rats to reduce the environmental variables so that they could have the results as most accurately as possible. Therefore, they were very controlling on how they maintained them. The rats were housed and cared under the supervision of the Animal Care and USE Committee of the National Chung Hing's University under the guidelines of Taekwon. Animals were caged individually in temperatures between 24 degrees Celsius, one up or one down. The humidity had to be between 60 to 65%. And they were on a light controlled room with a cycle of 12-12 light dark cycle, meaning that they were in complete darkness for 12 hours, and the other 12 hours they had light. The food and water was also controlled. They were all being fed the same food. This led to them, once they have selected their lab rats, they needed to get their FAB-induced rats. In order to prepare the FAB-induced rats, the scientists had to prepare them. They did this by infusing FAB into the brain to induce experimental Alzheimer's disease. The FAB-infused lab rats had to be induced for a total of 4 weeks before the scientists could go on to the next step. To make sure they kept it consistent, there was an osmotic pump implanted on the back of the rat. Um, the osmotic pump had a delivery rate of 2.5 microliters per hour for the 4 weeks. During this step, there was no harm done to the animals as they went under anesthesia to not feel a pain. All the animals survived and were able to go into the next step. In this section, well, the next step was that the scientists separated the lab rats into different categories. They had the FAB infused rats and their normal lab rats. The first category was 10 lab rats that were labeled normal group, which was going to be essentially used as a control group. The second group was 10 normal lab rats plus a high level of ATX. This was done to see if ATX was going to have an effect on the animals and to see if there was going to be an increase of its functions, such as awareness, memory, and speed, or if there was going to be a decrease. The third group was the FAB-induced rat. The rats got divided into 3 subgroups. The first one was a FAB-infused group with no ATX treatment group. The second one was FAB-infused and low dosage of ATX treatment group. And lastly, it was a FAB-infused and high dosage of ATX treatment group. This will allow the scientists to measure if the different amount of ATX would make a difference between the FAB-infused rats. Once the groups were completed, they could go on to the next step. In the next step, the scientists had to prepare the lab rats to go into the Morris water maze. The way that they would do this is they would focus on the ATX lab rats as they needed to be injected for a total of 7 days, regardless of their dosage. This would occur, they would get injected every day at 9 a.m. on the dot. The scientists would then take them, once they were on the last 3 days, to the water maze 1 hour after the injection of the ATX to evaluate their progress. As stated prior, the Morris water maze is designed to test spatial memory and long-term memory by observing and recording escape latency, distance moved, and velocity during the time spent in the Morris water maze water tank. The image to my right is how the maze will look like once assembled. This maze is looks like a pool compared to a corn maze where lab rats would have to go through this maze specifically, they would just be placed and they would have to swim uh, and get familiarized with their environment to be able to find the platform and escape the water maze. To explain it a little more, I have created a diagram as you will see on the next slide. Okay, so how does the Morris water maze work? So basically, it's assembled, water is placed, and then the rat is placed in the water. As we see in the first diagram on day one, they are um, they're expected to escape the water maze and to find the platform. On the first day, you will see that the lab rat goes around in circles and loops and around the edges because they don't know what they're looking for or where they're at. They need to get familiarized with the environment. So they had to keep going and going until they finally find the platform. On day number two, they already know and have the expectations of what it somewhat looks like. Therefore, they don't do as much uh, swimming around as they did compared to day one. Then on day three, that's when they already know their expectations. They already know where the platform is. Therefore, they can swim a little straighter compared to day two and day one. The rats were then tested on one trial per day, as we stated on their last 3 days of injections of ATX. Um, each rat was individually tested before the beginning of the test. The rats were placed on the platform for 60 seconds to familiarize with the environment and the location of the platform. To run the test, rats were randomly placed in different quadrants of the pool in each trial. Rats were allowed to remain on the platform for 60 seconds after succeeding to escape or fail to locate the underwater platform with 180 seconds. The escape latency of the lab rat were averaged and subsequently analyzed. The swimming distance and time spent in the target quadrants was calculated. The whole process was recorded with a video camera and analyzed with a smart video tracking system. The system would record escape time, swimming speed, and path lengths and directions. Data collected and results. Rats of all five groups spent shorter times to find the underwater platform starting on the second day of the third consecutive days of trials, but there was no significant difference among the groups. The escape time is located in panel A, while swimming distance is in panel B. The escape time on the third day follows with panel C, as we see on the bottom left corner, and the swimming distance on third day is the panel D on the bottom right corner. All this data was analyzed and plotted. Two-way ANOVA and AID show significant difference found between the different days of testing. For panel A, the x-axis consisted of average escape time in seconds in each day, and y-axis displayed the day and dates that they were collected for. In panel B, the swimming distance is on the y-axis and the days are on the x-axis, while panel C has the data collected on the third and final day. The y-axis has the escape time on the third day in seconds, while panel D has the swimming distance on the third day on the x-axis in the groups of labs there were. The results of the escape time on the third day indicated the FAB-induced rats spent more time to find the platform than the control rats. Low dosage of ATX treatment in FAB animals failed to reduce it. However, high dosage ATX treatment in FAB animals effectively decreased search time. During the trials, they made an observation that the FAB-induced rats seemed confused to their surroundings and that they had a tendency of going in circles. Another observation they came was that the normal was the high ATX group actually made better than just the regular normal lab rat. The results of the acquisition in the Morris water maze test displays that there was a difference in performance between the different groups in lab rats, including the control group. All the groups of lab rats shortened their time and to find the underwater platform starting on the second day of the 3 consecutive days of trial. The results of the escape time on the third day indicated that the low dose of ATX treatment in FAB-induced rats spent more time to find the platform than the ones with a higher dosage. The data then confirms that their hypothesis of ATX was right, and ATX can be a potential cure of Alzheimer's, or at least it can help diminish the symptoms of Alzheimer's and not allow it to progress as fast as it does. However, it must go undergo more testings as they need to verify that it is okay to be used in humans and to see if there are any potential side effects. Okay, and finally, we are here, guys, the take-home messages. The first message is that no one is immune to Alzheimer's disease. See a provider if there is any symptoms that you have. The earlier that it is detected, the better, as there are more treatment options, and you want to make sure that you're always updated with your information regarding the treatment and what options there are out there. The second take-home message is that animal models of disease are a cornerstone of the drug developmental process, as in this case, the lab rats have been a huge help in trying to find a cure for the Alzheimer's disease, and they have proven that ATX does help slow down the progress of the Alzheimer's disease and all the benefits that it comes with it. The final take-home message is that through science, we are getting closer to a cure. There just needs to be more research. There needs to be research in the sense that we need to know if it's okay to use in humans. Is there any other side effects of this? Are there short-term side effects, long-term side effects? Can this can ATX be mixed in with another medicine that's out there currently and make it 10 times better, 100 times better, or completely come to a cure? So that is it for today, guys. Thank you for listening in with me, and I really hope that you learned something through this presentation. Here are my citations. If you have any questions, any comments, feel free to leave under the comments. Thank you so much and hope you have a wonderful day. And I really hope that you learned something today.